Hello everybody, welcome to this video. In this video, we are going to do a quick little review of the concept of osmosis. Okay, so first off, I just want to start off with the definition of osmosis. Osmosis is the diffusion of water. So water, being a molecule, is going to behave like any other molecule would, in that it is going to move from an area of higher concentration to an area of lower concentration. So an area of higher water concentration to an area of lower water concentration. Osmosis typically happens across a selectively permeable membrane, meaning a membrane that lets some things through, but not others. Okay, so your plasma membranes of your cells, epithelia, those are selectively permeable membranes. Okay, so taking a look at this diagram here, what we have happening is we've got this solution here, selectively permeable membrane here, and a like a movable plunger here, okay? The purple circles indicate glucose molecules. And as you can see, we have a higher concentration of glucose on the left. Sorry, that would be the right. We have a higher concentration of glucose molecules on the right than we do on the left. However, since we're talking about osmosis here, what I want you to do is to take a look at this same situation, but from the point of view of water, right? So if you look at this diagram here, which of these compartments has a higher concentration of water? Okay, and then considering the fact that water is going to diffuse, because we're assuming that this membrane is permeable to water, which I guess I should have told you earlier, which direction will water move in? So just take a moment to think about that. Okay, so hopefully you figured out that water is at a higher concentration on the left than it is on the right. Therefore, water is going to move down its concentration gradient from the left to the right. And that is what is shown here in this diagram here. As we move water from the left to the right, that increases the volume on the right side and it moves this plunger, okay? All right, now I want to introduce this concept of osmotic pressure, okay? So osmotic pressure is the pressure necessary to prevent osmosis, right? So up here, we had osmosis happening. We had water moving from the left to the right. If we wanted to prevent that, we would have had to apply pressure to the plunger here on the right to keep that water from moving from the left to the right. So that is our osmotic pressure, is this pressure that we're applying here, okay? All right, now I want to move on to this concept that osmosis doesn't always occur. Sometimes we have osmosis happening, sometimes we don't have osmosis happening. How can we tell when osmosis is gonna happen or not? Basically, whether osmosis occurs is going to depend on the property of the selectively permeable membrane, okay? So to illustrate this concept, we have this figure on the top where we're looking at a cell in a beaker, okay? This cell has a cell membrane, which is selectively permeable. It's going to allow water through, okay? But it's not going to allow this solute through. So we have these green solutes with N's in them, okay? And the N stands for non-penetrating, but basically what it means is that it is a solute that is not able to cross this selectively permeable membrane. And then down here, we have a scenario where we have a cell with some solutes in it. In this case, these solutes can cross this selectively permeable membrane. So it's the same selectively permeable membrane, it just happens to be permeable to this solute here in purple, but not to this solute here in green. So what I would like you to do now is take a look at these scenarios and think about which molecules are going to move in each scenario and in what direction they will move, okay? And again, I just want to emphasize that water can easily cross the membrane through pores that exist in the membrane. So go ahead and hit pause to think about that, and then we'll come back to it. Okay, let's take a look at what has happened in these two different situations. Okay, so in the top situation, we had this non-penetrating solute, this green solute that is not able to cross the membrane here. Since it can't cross the membrane, what's going to happen is water is going to cross the membrane and it's going to move into the cell because it is at a higher concentration outside the cell than inside the cell, okay? And then that's going to cause this cell to swell. In the scenario on the bottom where we have a solute that is capable of crossing the membrane, that solute is just going to cross the membrane to go down its concentration gradient. So that's what happens here. We have this solute leaving the cell down its concentration gradient, and we don't have osmosis happening, okay? Because basically after that solute has crossed the membrane, we've evened out the concentrations on either side of the membrane. Okay, last concept that I want to go over here is osmolarity. Okay, so we have this term called osmolarity, which is a measure of how much solute we have in a solution. Okay, so the osmolarity of a solution can be calculated as the moles of particles per liter of solution. Okay, and that is that gives you a unit of osmolar. Okay, we're not going to do very many calculations of osmolarity, but it is important to understand what it represents. So that's what I'm going to go over here. So looking here at these two solutions, we've got A and B. Just to test you on this concept, which solution has a higher osmolarity? So go ahead, think about that for a second. Okay, so hopefully you determined that solution B has a higher osmolarity because it has a higher concentration of solutes, right? So higher number of moles of particles per liter of solution. So basically more solute means higher osmolarity, okay? Another thing that I'd like to talk about is that we use these terms, we have terms that we use to compare the osmolarities of certain solutions to each other, okay? So we have this term here, isosmotic, which means same osmolarity. We have this term, hyperosmotic, which means higher osmolarity, right? Hyper means more or higher. Iso means same. And then we have this term, hyposmotic, which means lower osmolarity. Okay, so hypo means below, under, less than, okay? And again, I just want to emphasize that these are relative terms. You would only use them to compare solutions to each other. You can't describe a solution as being hyperosmotic. It's a comparative term. It has to be described as hyperosmotic to some other reference solution, okay? So I have this exercise for you to think through, right? We're looking at solution A and solution B here. What words would you use to fill in these blanks? So go ahead and think about that for a few seconds. Okay, so solution A is hyposmotic to solution B. It has a lower osmolarity than solution B. Solution B is hyperosmotic to solution A. It has a higher osmolarity than solution A. So hopefully you figured that out And that basically wraps up our discussion of osmolarity, osmosis. Hopefully this was a good little refresher for you, and hopefully now you're ready to think about osmolarity as it pertains to processes in the human body.